Okay, so the plasma membrane is what regulates everything moving in and out of the cell. And that's important. We don't want just anything moving in, and we don't want just anything moving out. The cell needs to be able to regulate how that happens. So that plasma membrane is a boundary between the internal environment of the cell and the external environment of the cell. And it's what regulates everything moving in and out of the cell. It also is what receives stimuli from the outside and initiates a response inside the cell. And there are a lot of different cells in which that is what happens. So depending on what kind of cell we're talking about, there are going to be different structures in that plasma membrane that carry out the job of that cell. For that cell to do its cell job, it needs to be able to communicate with other cells, and it needs to be able to receive stimuli, and it needs different molecules to move in and out. How those molecules move in and out depends on what kind of molecule we're talking about. And today I'm going to give you some additional information that will hopefully make it a little more clear about what types of materials can just easily move across the cell membrane through that phospholipid bilayer, which molecules can move across pretty easily, but they have to have a protein to facilitate that diffusion. And then when we actually have to use active transport to move molecules in and out of the cell. So really kind of three types of movement across the cell membrane. Of course, all transport across the cell membrane is really divided into two major categories. We have what's called passive transport, which doesn't require any energy. And when we say no energy required, we mean no ATP required. So remember, ATP is the energy source for the cell. So anytime we say energy required, we mean ATP. So active transport then is the type of transport that does require energy. And again, when we say energy required, we mean ATP. And today we'll talk more about how to know when energy is required versus when things are just going to passively move across without any energy required. Any questions just about that part? Okay. So this is a pretty complex picture. It has a lot going on here, and I just want to point a few things out on this picture because the words are small and there is a lot going on here. So the first thing you should notice is that this cell isn't flat, okay? Cells are three-dimensional, and so you can really get a feel for that in this picture with that phospholipid bilayer really continuing back into the picture. It's not just that one layer of phospholipids. But you see that the cell membrane is mostly phospholipids. So remember that these heads that are pointing toward the extracellular fluid and the intracellular fluid, all the polar heads, the water-loving heads, are facing what's mostly water, okay? And then those tails, since the tails are hydrophobic, they're being repelled and they're all pointing inward. And that's how we get this, what we call a bilayer. So it's mostly a phospholipid bilayer. And that alone really has a lot of control over what's entering the cell. Because if you can imagine, we have this layer of non-polar tails. So if this whole region is non-polar, something that's polar can't just come across. So just those tails alone are really going to have a lot of control over what comes in and out of the cell just passively. And that's why we have proteins also embedded in the phospholipid bilayer, because those proteins are going to allow certain molecules to come across that couldn't normally come across. And we're going to talk about what molecules those are today. But right now I just want to point out really the structure of this plasma membrane, and then we'll talk about some of the functions coming up. So these proteins that are embedded are all purple in this picture, and you can see that some of them span the entire distance. So they have part of the protein is actually interfacing with the extracellular fluid, and part of it is actually interfacing with the inside of the cell. So this part of the cell is called the cytoskeleton that you learned about in the previous videos last week. The cytoskeleton is interfacing with those proteins, and also the intracellular fluid. So these are called transmembrane proteins. The transmembrane proteins really are the proteins that span the entire distance. So trans means across. So this means across the membrane. The transmembrane proteins are going to interface with the outside of the cell and the inside of the cell. And extracellular fluid is usually abbreviated ECF. And intracellular fluid is usually abbreviated ICF. And those are very important terms to know when you're talking about the cell. So the extracellular fluid is everything outside the cell, and the intracellular fluid is all the fluid inside the cell. And that's really where the chemistry of the cell is happening, is in this intracellular fluid. And then of course, it's being affected by what's out here. Some of the proteins don't really span the entire membrane. So we have some proteins that are just kind of sitting out here. So let me draw a couple more phospholipids here on our membrane. If we had a protein that was tucked in here and it didn't span the whole membrane, that's called a peripheral protein. So a peripheral protein can be just on the outside. It can also be just on the inside, but they're mostly on the outside. But you can see in this picture that we also have peripheral proteins on the inside of the cell. But we usually have a lot more on the outside. So those peripheral proteins are going to receive a signal and do something. They could also be enzymes. So this peripheral protein could be an enzyme. So it's going to cause some type of reaction to take place. When that substrate binds to the active site of that enzyme, it's going to catalyze that reaction. And it could also be some type of receptor protein, where a certain molecule will bind and it will cause a certain response to happen. So there are some proteins that span the whole distance, and those are called transmembrane, and then we have some just on either just the outside, or we could have one just on the inside, and that would be called a peripheral protein also. Any questions about that? Okay, and then you see attached to some of these proteins, we have these carbohydrate chains. And these carbohydrate chains are very, very important to the cell because these are how the cell recognizes self. Cell recognition is carried out by these, they're called either glycoproteins or glycolipids, depending on if that carbohydrate chain is attached to a lipid or if it's attached to a protein. So here you see a chain of carbohydrates. It looks like just a chain of glucose. It's in green on this picture, and it's just attached to a lipid. So that's called a glycolipid. It's just attached to one of the phospholipids. If it's attached to one of the proteins, it's called a glycoprotein. And in the lecture videos, I talk about a really important glycoprotein called the MHC. So the MHC stands for major histocompatibility complex. And this is really the most important glycoprotein that you have on the surface of your cells, because this is how the immune system recognizes that those are your cells. So this is self-recognition. So if you got any type of tissue put in your body that wasn't your own, your immune system would recognize that that MHC wasn't yours, and it would start attacking and destroying that tissue. So when people have any type of tissue or organ transplant, they have to go on really strong immunosuppressant drugs in order to keep their body from fighting off that tissue. And that's really the role of your MHC, which is one of your glycoproteins. Okay, we also have glycoproteins on our red blood cells that are involved in ABO blood typing, and those are different glycoproteins. So you can have a type A marker on your red blood cells, or you can have a type B, or you can have both. And we have a blood typing lab coming up where you'll learn about that in more detail. So that's what those chains of carbohydrate are on the outside of the cell. Those are cell recognition markers that help our immune system recognize self. Any questions about that? Nope. Okay, and then lastly, the last one I want to talk about is cholesterol. Cholesterol in the cell membrane is incredibly important, and we have a whole slide coming up that's just going to talk about cholesterol. Cholesterol is really going to play an important role in stabilizing the cell membrane, and it plays a role in permeability, meaning what can come across and what can't come across. And we do have a slide coming up about that. So that's the basic structure of the cell membrane. It is more complex than just phospholipids. Sorry, it's not wanting to go to the next slide. Okay, when we're talking about the phospholipids, so we have this head region, of course, that's polar, and it's made mostly of a phosphate group. It also has a glycerol alcohol and this part called a choline group. But what you should notice about this picture is that one of the hydrophobic tails is a straight chain, and one of them has a bend in the chain. So for this, you have to think back to the lipids lecture. And remember that we have two types of fatty acids. We have saturated fatty acids, and we have unsaturated fatty acids. One of these is a linear chain of carbon and hydrogen. So remember, they're both hydrocarbon chains, meaning they're a chain of just hydrogen and carbon. They do have, if you'll remember, a carboxyl group also, but otherwise it's a carboxyl group plus a hydrocarbon chain. One of these is linear, and one of them actually has a bend in the chain, especially if it's a correct configuration called the cis configuration, then we know that it has a bend in the chain. Okay, so now this is going to be our first practice at using the chat feature for participation points. Again, you won't be judged on your answer, and today I am still taking attendance the old way, but I want everyone to practice doing this. Okay, so the question is, which type of fatty acid chain has the bend in the chain? Is it the saturated fatty acid or the unsaturated fatty acid that has the bend in the chain? So you'll just type your answer into the chat. Awesome. Well, you're all correct. It's the unsaturated fatty acid. Okay, so it's important to know that phospholipids can have either. Okay, so a phospholipid can have either a saturated or an unsaturated fatty acid. It can have one of each, or it can have two of the same. What this does to the plasma membrane is when there's this kink or this bend in that hydrophobic tail on this phospholipid, it creates a space in the plasma membrane. And this is how small molecules are allowed to just simply diffuse across the membrane. So one of the ways that simple diffusion happens is if we have in the phospholipid bilayer, so we'll do a straight one and then we'll do one with a kink. Okay, and so then over here, we have another one with a kink. That's going to create a space in the phospholipid bilayer that's going to allow small, uncharged molecules to simply diffuse across through simple diffusion. And remember that the way diffusion is going to take place is from an area of high concentration to an area of low concentration. And of course, this would be a bilayer. We'd have the phospholipids on this side too. But that bend in the chain is what is allowing that space for simple diffusion to occur. So depending on what kind of cell we're talking about, one way that a cell can increase permeability, in other words, have more molecules coming across through simple diffusion, is to have more of these unsaturated fatty acid tails on the phospholipids. And in human cells, most of our cells actually have phospholipids with one of each. Again, depending on what type of cell we're talking about. If you have a cell where most of the phospholipids are saturated fatty acid, there aren't many spaces. That cell would be pretty impermeable. I don't know why this isn't switching. Okay, so one of the ways where molecules can get across the plasma membrane is these gaps in the phospholipid bilayer. And again, what causes those gaps is this bend in the chain or the unsaturated fatty acid tails on that phospholipid. Another way molecules can get across is through the proteins. And remember, if a protein is involved, it's not called simple diffusion, it's called facilitated diffusion. So I'm not going to repeat everything that's in the videos. If you haven't watched the videos yet and you don't know the difference between simple diffusion and facilitated diffusion, it's very, very important to watch those videos. This does not replace that, okay? This is in addition to that. So you still need to watch those videos to understand what I mean by simple diffusion versus facilitated diffusion. So simple diffusion is going to be molecules moving from high to low concentration, and they're just moving through gaps in that phospholipid bilayer that are caused by the kinks in the tail. Okay, then transmembrane proteins are going to be involved in facilitated diffusion. Transmembrane proteins are also involved in active transport. So when we say transmembrane proteins, we're talking about those proteins that span the entire distance from inside the cell to outside the cell. Okay, and then lastly, the third way that molecules get across is through endocytosis and exocytosis. And that involves vesicles. Remember, a vesicle is a sac of membrane. So it's just a sac made of phospholipids, carrying a molecule, carrying a cell product. So carrying a cell product from inside the cell to outside the cell. And then when another cell accepts that product through endocytosis, it's wrapping that product in membrane and bringing it in. So that's going to be the third way that we bring materials in. Okay, this shows you a better picture than what I drew on the board. membranes with gaps that would allow simple diffusion to come in. So you can see not many gaps here when everything is just saturated, but we do have gaps in the membrane when we have those unsaturated fatty acid tails. Okay, the other thing that allows molecules to move across more easily is cholesterol. Okay, so it's the opposite of what you would think. Cholesterol actually stabilizes the membrane. So the more cholesterol you have in a cell membrane, the less permeable it is. Okay, so permeable means materials can move across. And the key phrase that's typically used when we talk about the plasma membrane is we say that the plasma membrane is selectively permeable. In other words, the plasma membrane is selecting what's allowed to just come across. And the way it selects is through the chemistry and the structure of that plasma membrane. And one of the things it can do structurally is if there is more cholesterol embedded in that membrane is very stable and doesn't allow much simple diffusion to occur. So the less cholesterol you have, the less the less stable that membrane is and the more permeable. So it's kind of the opposite of what you would think. So one of the ways you can make a membrane more permeable is just have less cholesterol stabilizing it. If you want it to be less permeable, you'll have more cholesterol. So you can see the cholesterol here, this isn't allowing those gaps to occur. It's really stabilizing that membrane. So more cholesterol makes it less permeable. Less cholesterol makes it more permeable. Any questions about that? Okay. Oh goodness. I think it's anytime I click on something else. Okay, here we go. The proteins that are embedded in the cell membrane, we've already talked about transmembrane versus peripheral proteins, but even just in the category of transmembrane proteins, there are several different kinds that you should know about. And really, you don't need to know all of these types of proteins. I really wanted to show you that there are more than what we're going to talk about in this class. In this class, you really just need to know about channel proteins versus carrier proteins. And we're going to talk about the differences between these two. But this picture kind of is a sneak preview. When you take physiology, you will learn about these additional types of proteins. Okay, and then notice that this one here, cell identity marker, has a carbohydrate on it. So this one would be called a glycoprotein. And this is how cells identify each other. And this is how your immune system recognizes self, is through one of these transmembrane proteins that has this carbohydrate attached. This slide is straight from the lecture video on YouTube. So if you've watched those lecture videos on transport across cell membranes, you've seen this slide, and it really is just a reminder of the different types of passive transport versus what active transport is. So remember, if it's passive transport, that means all of those molecules are going from area of higher concentration to area of lower concentration. So in this picture, the ECF, the extracellular fluid, has a higher concentration of all three of these solutes compared to the inside. So everything's going to move from outside of the cell to inside of the cell. So from high concentration to low concentration. If it's just moving across the phospholipid bilayer with no proteins involved, that's called simple diffusion. So simple diffusion does not involve proteins. So no proteins involved in simple diffusion. It's just going across the phospholipid bilayer. Okay, but if a protein is required to get that molecule across, because it can't just diffuse across those gaps in the phospholipids, then we call that facilitated diffusion. So facilitated diffusion is going to require proteins, and it's going to require one of those two categories of proteins. It's either going to be a channel protein or a carrier protein. Oops, I said protein and I wrote facilitated protein. Facilitated diffusion. So still going high to low, still not requiring energy. So it's still passive transport, but because a protein is required, then it's called facilitated diffusion. The proteins are facilitating that diffusion. Those molecules can't just go across the phospholipid bilayer. A channel protein, the name really says it all. It's a channel that these molecules or ions are going to pass through. And we have a lot of ion channels in our plasma membrane that allow ions to move in and out of the cell along a concentration gradient. No energy required, just moving from high to low. A carrier protein involves a molecule binding to the surface of the protein, and then the protein changes shape and releases that molecule to the inside of the cell. So rather than just having a free flow back and forth, it's going to just open up and allow that molecule into the cell. It's actually carrying it in. So I guess it's kind of like, rather than walking through a tunnel, the molecule has to sit down in its seat and be transported like a bus into the cell. Active transport, on the other hand, requires ATP because we're going against a concentration gradient. Okay, so active transport is going the opposite direction. With active transport, the molecules are going from low concentration to high concentration. So high concentration, I'm just going to abbreviate it, CONC, to low concentration. So we say that's against a concentration gradient. It's like pumping uphill. It requires energy. If we want water to go downhill, it just flows downhill with no energy required. Okay, in fact, it produces energy as it's flowing downhill. But to get that water back uphill, you have to pump it, and that's going to require energy. The same thing goes for the cell. To go against a concentration gradient is going to require energy in the form of ATP. So ATP required. So there are times where you need all of a certain molecule out of the cell, or all of a certain molecule into the cell. And you can't just count on concentration gradients, because in diffusion, whether it's simple diffusion or facilitated diffusion, the goal is equilibrium. So let me get to that slide. Okay, so in passive transport, if we have all of these molecules outside the cell, they're going to start moving from high to low. So they're going to start moving into the cell until they become equal on both sides of the cell. So the goal of passive transport is equilibrium. It's not really even the goal, it's really the end result. So the end result of passive transport is equilibrium. A state of being equal. So we want the molecules to be equal on both sides of the membrane. So in the ECF and the ICF, they would be equal. We don't always want them to be equal. Okay, so if I wanted to get all of these into the cell or all of these out of the cell, I can't just count on simple transport. I'm going to have to pump. to get these out. Okay, I'm not just counting on them diffusing across. So if I wanted to get all of these guys into the cell, at some point I'm going to have to move them in, and that's going to require energy. We're going against a concentration gradient. So the end result, or we can say the goal, of active transport is not equilibrium. Any questions about that? I'm actually going to quickly go turn off this light overhead because it's really, I don't like the glare on the board. I'll be right back. Oh, I still have the glare. Okay, so the glare is actually, it's the sun. I can't do anything about that right now. Okay, so when we're talking about simple diffusion and not requiring a protein, so things that can just move across the phospholipid bilayer, this is a list of who can just move across. So oxygen and carbon dioxide. So if you watched the lecture videos on YouTube, you know that the way we do O2 and CO2 exchange between the air sacs in our lungs and our body cells, and getting that oxygen to the cells and getting that CO2 back to the lungs to exhale is just done by simple diffusion, everything moving high to low. Okay, so what that means is if you want oxygen to get to your cells, you have to have a constant supply of oxygen in the bloodstream, and that supply of oxygen in the bloodstream needs to be higher than the concentration inside the cell. Okay, it also means you have to be exhaling so that the concentration of CO2 in your blood is less than your cell. Otherwise, that CO2 won't move out of your cell. CO2 is very toxic to your cell. And the whole reason that we breathe in oxygen and breathe out CO2 is cell respiration. So remember the general formula for cell respiration. For every 1 glucose that comes into the cell, we need 6 oxygens to combust that glucose to produce ATP. And as a product, we're going to make carbon dioxide and water. And on average, 36 ATP. This CO2 is very acidic. We don't want that sticking around. Okay, we want to get it to the air sacs in the lungs and exhale it. And in order for that to happen, it needs to be lower in the blood than the body cells, so it will diffuse from the body cell into the blood cells. And the blood cells will carry it back a little bit, and then a lot of it is just dissolved in your blood. So CO2 can either be dissolved in your blood or it can be carried by your red blood cells. It's mostly dissolved in your blood. So that's simple diffusion, O2 and CO2 exchange. Okay, sometimes water can move across. Now, this seems kind of counterintuitive because those nonpolar tails actually repel water. But if you have a cell with a lot of unsaturated fatty acid tails in their phospholipids, there are some gaps where water can slowly, passively diffuse. But most water actually comes through proteins called aquaporins. And we're going to look at those in a minute. This means water pores. So we do have channel proteins that actually allow water to move in and out through osmosis. But a little bit of water can sneak through those holes. It's a little bit leaky. Okay, really small uncharged molecules can move across. So things like, and you know, we do an experiment with the cell coming up, and iodine can easily move across because it's super small. So it can move through those gaps. It's not charged, and it can easily move across. Remember, this uncharged is important because remember that ions have a charge. So things like sodium, potassium, chlorine, calcium, those can't just move across the plasma membrane. Those are going to need proteins. Steroids and fatty acids, those are both non-polar. So steroids would be any of those steroids made from cholesterol, and then obviously fatty acids. Why can those just move across? Those can move across because they have the same non-polar composition as those fatty acid tails, so they're going to be allowed across. Anything else is going to need a protein to get in. It can't just cross that phospholipid bilayer. So any questions about this list of what can just simply diffuse across? Okay, so what cannot just diffuse across? Anything with a charge. So there are molecules that are charged, and then there are obviously ions that are charged. So an ion would be things like sodium, potassium, chlorine, calcium. Those can't just move across passively. Those are charged. They would be repelled by the nonpolar tails of the phospholipids, so those have to come through a protein. Really large molecules, such as glucose. Glucose is too big to just come across the phospholipid bilayer. It must have a protein to get into the cell. In fact, all of those big monomers that come from our food have to come through a protein to get into the cell. Okay, and then anything that's polar, obviously nonpolar and polar repel each other, so anything that's polar can't just come across those phospholipids. Any questions about that? Okay, again, this is all additional material. It is not in the lecture videos. This is just a picture of the aquaporins. So this is a channel protein that only allows water. So channel proteins and carrier proteins are both very specific in what they'll allow across. So an aquaporin will only allow water. A sodium channel will only allow sodium. They're very, very specific in what they'll allow in or out. Okay, that things can move both directions. Water can move in or out. Sodium can move in or out, depending on the concentration gradient. So I mentioned this earlier when I was talking about cholesterol, but now this really ties in with the aquaporins also. Water, if you watch the videos, you know that water moves toward the higher solute concentration. So if you can't move the molecule, you can move water. So if we had a cell, and let's say it has a 5 molar solute concentration inside, and this is total solutes. So it's the total of everything. I'm not even going to list an individual solute. This is all the solutes. Let's say the concentration is 5 molar. Okay, and let's say out here it's 1 molar. So this is the ECF, and this is the ICF. Okay, extracellular fluid and intracellular fluid. Looking at the ECF, we're going to do another vote by chat. Okay, looking at the ECF, is that ECF hypotonic, hypertonic, or isotonic? Now, again, we're talking about the ECF. So if everyone can vote through the chat for if you think it's hypotonic, hypertonic, or isotonic. And again, you won't be judged on if you get it right or wrong. Okay, only one person has voted so far. Here we go. Okay, remember hypo means below. So if you have hypothermia, it means your body temperature is below normal. If you're hyperactive, your activity level is above normal. So hyper means above, and iso means same. Isometric exercises mean you do the same on each side. So ECF 1, ICF 5, 1 is below 5. Okay, so we would say this is a hypotonic environment for that cell. And in a hypotonic environment, okay, water is going to move toward the higher solute concentration. So that means water is going to move into the cell to dilute that solute concentration. Okay, so if you can't move the solutes, then you can move the water. That water is going to go through the aquaporins. If we wanted to reduce the amount of water, okay, if we wanted this cell to have a higher solute concentration and we didn't want water to just passively come in, a couple of things we could do. One is to just not have that many aquaporins in this cell. So the fewer aquaporins you have, then the less permeable this cell is going to be to water. Okay, then the other thing we can do is we can put more cholesterol in that membrane. If you have more cholesterol, then that cell is also less permeable to water. It's less permeable to everything. So more cholesterol and fewer aquaporins, and we can really regulate water coming in and out of the cell. Any questions about that? So again, aquaporins, that's just the name for those channel proteins that carry water in and out of the cell. A little bit of water can leak across, but it really mostly goes through those aquaporins. Okay, these are just some examples of ion channels. Again, these are channel proteins and they are very ion specific. So we have sodium channels, calcium channels, potassium, chlorine. Moving these ions in and out of the cell is extremely important to your nerve cells and your muscle cells. The way you conduct an electrical signal through your nerve cells is by using these ion channels to move certain ions into the cell or out of the cell. And we're going to talk about that in more detail in a minute. You also sometimes have to pump ions to get a difference in charge across the membrane. We're going to talk about that also. We actually have more potassium channels in our cell membranes than any of the others. You don't really need to know that right now. If you're curious what we have more of. You don't need to know this level of detail right now, but I just wanted to show you that some ion channels are always open. This is just a common question that students ask, so I decided to add this slide. Some channels are always open, but some channels only open when a certain thing happens. Okay, so this first example, you don't need to know this term ligand gated, but this channel only opens when a neurotransmitter binds to the channel, and then it will open. Okay, this one is what is called mechanically gated. It only opens when pressure is applied to the plasma membrane. Okay, and then voltage gated, when the voltage reaches a certain level, then the channel will open. So some channels are always open, but some open by other means. Okay, the sodium-potassium pump is really one of our best examples in this class of active transport. And it's important to understand just the very basics of the sodium-potassium pump as an example of active transport. So remember, active transport is going to require energy, and we're going from low to high concentration. So we're going against a concentration gradient. So you can write out sodium, potassium, or you could just do the short version, which is just showing the ions like that. Okay, and this is active transport. Okay, I'm going to start by showing you a little video of the sodium potassium pump, and then I'm going to talk about it in more detail. Okay, I have to stop sharing for a minute and do a new share to get this to play, sorry. Oh, okay, sorry, here's what I'm going to do is this.
Active transport is a type of cell transport that requires the input of energy in the form of ATP. The proteins that conduct this form of transport are often called pumps because they force molecules or ions to move from an area of low concentration to an area of high concentration. This is commonly referred to as against the concentration gradient. One of the more common examples of active transport is the sodium-potassium pump. The job of the sodium-potassium pump is to move sodium ions, or Na+, out of the cell, and potassium ions, or K+, into the cell. Let's take a closer look at how this happens. The sodium-potassium pump has binding sites with 3 sodium and 2 potassium ions. Let's follow the movement of the sodium ions first. After 3 sodium ions are positioned within the carrier protein, an ATP molecule is split, releasing phosphate. This phosphate binds to a location on the exterior of the carrier protein, causing the protein to change shape. As the protein changes shape, the 3 sodium ions are released to the other side of the membrane. Next, 2 potassium ions position themselves within the carrier protein, causing it to undergo another change in shape. In the process, the phosphate molecule is released.
Okay, so I want to point out that there's not much potassium outside the cell. We're trying to get all of it that we can into the cell. So it's going from low to high. If we just opened up the potassium channels, so we do have potassium channels and sodium channels in this membrane, if we just open up those ion channels, then those 2 ions are going to reach equilibrium, and they're going to be equal on both sides of the membrane. But right now, we want all of the sodium out of the cell and all of the potassium into the cell. And in order to do that, it's going to require energy, and we're going to have to pump it. You can see that for every 3 sodiums that leave, 2 potassiums come in. So it's not an equal number. And there is a reason for that. There's a reason that's occurring. And you'll learn about that a little bit more today, but you'll learn about it a lot if you take physiology, because this is how we're going to set up an electrical charge across the membrane. Okay.
Once the phosphate molecule is released, the carrier protein expels the potassium ions into the interior of the cell. The carrier then resumes its initial shape, completing the cycle. Notice that for every 3 sodium ions leaving the cell, 2 potassium ions enter. Since both sodium and potassium ions have a positive charge, this unequal movement causes an electrical gradient to develop across the plasma membrane of the cell. A number of cellular processes, including the generation of nerve impulses, use this electrical gradient.
So it's really, really important, especially in nerve cells, to be able to cause this difference in charge across the membrane. And really the most simple version of this goes like this. So if you have a cell, and we'll make it a nerve cell. Okay, so nerve cells are shaped a little differently than some other cells. So this would be the big cell body. And we have all of these sodiums and potassiums, and they're both positively charged, but what it doesn't show you is that we also have a lot of negatively charged molecules, including some proteins in here that have a negative charge. So let's say that I'm going to make everything negative in red. So we have all these negatively charged ions, such as chlorine, and then we have some proteins in here that are all negative. Okay, and then we have some negatives out here. But we also have a lot of sodium and potassium. So let's say we have a few sodiums in here, a lot of sodium out here, because potassium pump's been working, so it's been getting sodium out of the cell. I'm sorry, sodium potassium pump's been working. So we have a lot more sodium right now outside the cell than we do inside the cell, but we still have some inside the cell. Okay, we have a little bit of potassium still out here. We have a lot inside. If we send all three of these sodiums out through one cycle of the sodium potassium pump, and we only send 2 potassiums back in, okay, that's a net loss of 1 positive ion out of the inside. And we already have these negative charges in here. What that's eventually going to do is make the inside of the cell more negative and the outside more positive. Okay, so if we were to look on both sides of the membrane, we would see, you know, right along the membrane here, we see on this side, it's more negative relative to outside, where it's more positive because of all these sodiums we've pumped out. So even though we do have some positive ions still in here, it's more positive outside compared to inside. This is a difference in electrical charge across the membrane. And that's going to be really significant when you start learning about how nerves conduct an electrical signal, how that action potential happens, how resting potential happens. So the sodium potassium pump plays a very important role in that. And this charge across the membrane, we call that membrane potential. So membrane potential is a difference in electrical charge across the membrane. In other words, on both sides of the membrane, it's different. Okay, let's see, question. Oh, no, that was just a late hypertonic. Any questions about that? I know it's a little complex compared to some of the other concepts. I'm going to go back to the PowerPoint for a second. Oops. Sorry. It's taking me back to the same place. Hold on. Sorry. Oh, goodness. There we go.
They're smart, and then they're street smart, like a hybrid with an available EPA estimated combined fuel.
Okay, hi. Sorry about the Ford Escape ad. Okay, so this slide is just a really good summary of the sodium potassium pump, and I know it's really small, but it does require ATP. It requires 1 ATP for every cycle of the pump. And so for every cycle of the pump, we use 1 ATP, we pump out 3 sodiums, and we pump in 2 potassiums. So you can see in this picture, 3 sodiums bind. Okay, no one knows exactly what causes these 3 sodiums to bind. It's one guess is that there must be some negative charges in this point of the carrier protein. So this is a carrier protein, right? It's not a channel. So the sodium's not just moving through a channel, and the potassium's not either. They have to take a seat on the bus to get carried to the other side. So it's assumed that they're attracted by something negative in these binding sites. Okay, they bind, and now phosphorylation takes place. You're going to learn in detail what phosphorylation is coming up pretty soon. But phosphorylation, it happens when this ATP transfers a phosphate group to this molecule, giving it energy to do something. And what it does with that energy is it changes shape. When it changes shape, it releases those sodiums outside the cell. And now that those sodiums are released, it exposes these seats on the bus that perfectly fit the potassium. So now the potassium can bind. And when the potassium binds without any energy required at this point, the molecule still has energy from that phosphate group. Now the potassiums are going to get released inside the cell. To start the process again is going to require another ATP to transfer that phosphate group to the molecule to cause it to happen again. Remember that the test is multiple choice, so you don't have to explain this to me, you just have to know what happens. So if you have questions about that, be sure to ask, because there's at least one question on the test about the sodium potassium bond, maybe even 2. And it will be multiple choice. Okay, I'm not going to show this video on electrochemical gradient. It's pretty complex, and I don't think you need to know that right now. And there aren't any specific test questions about it. Just know what membrane potential is, and know that active transport is required for your nervous system to work. For nerve cells to function and transmit an electrical signal, active transport is going to be required to set up that membrane potential. Okay, it's important also to realize the factors that affect diffusion rates, and you're going to look at some of these in the upcoming lab. So next week's lab demonstrates osmosis and diffusion. And it does it pretty well. I like the lab. It is a difficult lab. Again, I sent out an email saying you need potatoes for next week. So don't forget to get your potatoes. You'll need those before you can start the experiment. You also need a knife, a cutting board, probably like 4 paper towels. And if you have a vegetable peeler, that's great. If you don't, then you can use a knife, but you do have to peel your potatoes. So 3 things that really affect the rate at which diffusion takes place. Okay, one of those is temperature. And if we were in lab, we would be doing a really specific experiment on how temperature affects diffusion rate. But the idea is, as temperature increases, I'm just going to abbreviate it as temp. As temperature increases, what happens is the kinetic energy of those molecules increases. Okay, they move around more. If you've ever lived in a major city where everyone just puts their trash out on the curb, you know that in the summertime, when it's hot outside, the trash is stinkier, right? And that's because all those stinky molecules are moving around faster in the air versus when it's snowing out, not only is the stinky stuff frozen, right, but the molecules aren't moving around as fast, so you don't have as much odor. So as temperature increases, kinetic energy increases, which means molecules are moving faster. Oops, right, energy twice. So molecules are moving faster. You can assume if molecules are moving faster, that means diffusion rate is increasing. So increase diffusion rate. In other words, as temperature increases, diffusion rate increases. Any questions about that? Okay, the other thing that really affects diffusion rate is molecule size. So bigger molecules actually diffuse much slower than small molecules. So this is kind of an inverse relationship. This would be a direct relationship. As one increases, the other one increases. Okay, big molecules are going to be more sluggish, and so large molecules diffuse slower. So in other words, as size increases, diffusion rate decreases. It's what we call an inverse relationship. Okay, and then that third factor affecting diffusion rate is difference in concentration across that membrane. In other words, the size of the concentration gradient. So you can imagine that if we had a situation I'm going to give you two scenarios, two different cells. And I'm just going to use dots for the solute. Sorry, I lost my red. Here we go. Now, I don't even know if you can see those dots on my camera. Okay, this one has a lot of red dots outside and only two inside. Okay, they're pretty closely equal. This one has a few more outside. Okay, this diffusion rate is going to be pretty slow. There's not much of a difference in concentration here. Okay, small difference here means slower diffusion rate. Okay, these are going to rush into the cell because there are a lot more outside than inside. So this is going to be a much higher diffusion rate because there's a bigger concentration gradient, bigger difference in concentration between the outside and the inside of the cell. This is going to be pretty slow. It's almost at equilibrium already. Okay, it's going to be just a couple coming in, maybe a couple more will go out and then a couple more in. But this will be pretty slow compared to this one. Okay, any questions about that? Okay, the very last thing is I have one more question for you. This is a practice question. Let me move this over. Okay, so over here on the left, I have passive transport, and here on the right, I have active transport. Okay, and I'm going to have you vote on each of these terms. We're going to put all of these terms that are up here. So I have 8 terms up here, and we're going to put all 8 of those terms in one or the other, or there could be a term that applies to both passive and active. And we're going to, so it's either going to be passive, active, or both. So I am going to have you vote on each word. So let's start with simple diffusion. Is simple diffusion passive, active, or both? Great, yep, that's correct. So it is passive. So simple diffusion, we're going to put here. Oh goodness, I have fat fingers today. Okay, endocytosis. Okay, now we didn't talk about that today, but it is in the lecture videos and hopefully you watch those. Endocytosis is how we bring in really big molecules and even whole entire cells. Does that require energy or does it not? So in other words, is it passive or active? Yes, it is active. That's correct. So most of you got that right. So endocytosis requires energy. It's wrapping membrane around. It's actually a pretty big energy requirement. Okay, channel proteins, where molecules just freely move through the channel. That channel, you know, at some point is open for that molecule to move in and out. Would that be part of passive or active? Yes, that's right. So channel proteins are going to be passive. Okay, osmosis. Osmosis, of course, is water movement. Is that passive or active? Yeah, you guys have got this down. So this is passive. So water movement does not require energy. We don't ever pump water in human cells. Okay, sodium potassium pump. Yep, sodium potassium pump, very active. It requires ATP. We're pumping against a concentration gradient. In fact, anytime you see the word pump, you can assume that pumps require energy. Okay, so if we had a pump at home, it would require some type of energy. And so sodium potassium pump is active transport. Okay, facilitated diffusion. Facilitated diffusion. Okay, I got a couple of actives in there, but facilitated diffusion is passive. Okay, so it's passive transport is going from area of high concentration to low concentration. It just means it has to be facilitated by a protein. It can't just go across the phospholipid bilayer. So facilitated diffusion. Oops. Okay, exocytosis. Okay, awesome. So just as it requires energy to move giant things into the cell, it also requires energy to move big products out of the cell. So if this was a cell producing insulin that needs to get out to the bloodstream, insulin production and release into the bloodstream from that cell is going to require energy for exocytosis to occur. Okay, and then lastly, carrier protein. Yay, Jenna, it's both. Yeah, a few of you are starting to get that now. It is both. That one requires some thought. Okay, so we can use a carrier protein in passive transport to go from high to low, but we can also use a carrier protein in active transport. And in fact, that is the main way that active transport occurs. So carrier protein. Sorry, I'm so bad at typing. My computer's at such a weird angle that I just can't even reach my keyboard. The carrier protein would go in both categories. Awesome job, everyone. Thank you so much for being here today. If you have questions, you can ask now, or I will, of course, be at my office hour today. And I will, as soon as we get off the meeting today, I will go on and post that link, because you do have to go into a different room now for my office hour so that the STEM Center has a faculty member available if other students have questions. And I will just put that right at the top on Canvas. So thanks, have an awesome day, and I will see you all soon. Bye.
Thank you.
Thank you. Thanks, everyone.